During the cell signaling devices, some of the proteins they play an important role, and one of these is the G proteins, which are coupled with the different receptors, and they make the cell to communicate with each other. Now, G protein coupled receptors, which are abbreviated as GPCRS, are so named because they interact with G protein. ये डेफिनेशन बिल्कुल सादा है, सिंपल है, क्योंकि जी प्रोटीन जो हैं ये रिसेप्टर जो हैं वो जी प्रोटीन के साथ एक्ट करते हैं, तो दे आर नोन एज जी प्रोटीन कपल्ड रिसेप्टर्स. नाउ देयर आर 7 ट्रांसमेम्ब्रेन receptors हैं, which are G protein receptor, and the member of GPCR super family are also referred to as 7 transmembrane 7TM receptors because they contain 7 transmembrane helix. 7 जो हैं वो transmembrane helix जो हैं वो उनमें मौजूद होते हैं, and they are known as the 7TM transmembrane. Now, for example, in this you see there are ligands which are there, receptor, amino proteins, carboxyl group, and they treat and go to the GTP or GTP and the G proteins which are there, जो कि नीचे हैं in this molecule, and there is a membrane. Further, they go to the effector and intracellular second messengers, they are produced which make the signal, and that is they are the G protein or the transmembrane receptor जो कि इस डायग्राम में शो किए हुए हैं. Similarly, this model depicting the activation of GPCR जो है by the X-ray crystallography structure वो शो किया हुआ है. जी प्रोटीन क्लस्टर जो है वो आप देखेंगे कि दैट इज़ एट द बॉटम. किस तरह वो जो मेम्ब्रेन है उसके साथ वो इफेक्ट कर रहा है. देन देर इज़ ए प्रोटीन प्लग्स विच आर एज ए मैटर ऑफ फैक्ट making the streams or channels through the membrane, and the signaling is passing from these channels to the cell exterior and the cell interior. और ये एक जो मॉलिक्यूल्स हैं उनको ये completely regulate करते हैं, उनको कंट्रोल करते हैं. We will see that there are natural ligand binding molecules to GPCRS, and that is वो कौन कौन से हैं. One is the diverse array of hormones, जो कि हार्मोन्स हैं both plant and animal, natural ligand, natural chemicals. Similarly, neutron transmitters, opium derivatives, chemo attractant, that is those molecules that attract phagocytic cells of the immune system, or also odorants and tastants molecules. They are detected by olfactory and gustatory receptors, यानी कि टेस्ट और स्मेल के जो हमारे ऑर्गन हैं उनके ऊपर जो इफेक्ट करते हैं, एंड eliciting the senses of smell and taste. And last is the photons, that is the light, जो कि light affected जो हैं वो हैं. Now in this table, the different stimuluses, receptor, effector, and the physiological responses of various organs that has been tabled for the ligand that operate. For example, you can see that with the light, the rhodospin, that is actually the visual action of the cell that they respond to that, and so on and so forth. All these different stimuluses that has been tabled and indicate the effector which is required. For example, so you see for the light rhodospin, you get cyclic CMP phosphodione and etc. so on and so forth. Now the topology of the cell is also very important, and we will see that the G protein coupled receptors, they normally have a topology where amino terminus, जो कि amino terminus of an amino acid NH2 is present on the outside of the cell. Cell के बाहर होता है उसके, और the 7 helices that traverse the plasma membrane, they are connected by loops of varying length. मुख्तलिफ लंबाई के loops जो हैं उनसे वो जुड़े होते हैं, and the eventually जो carboxyl terminus है amino acid का, that is present on the inside of the cell. Cell के वो अंदर होते हैं. The 7 helices that traverse the plasma membrane are connected by loops of various length, and the carboxyl terminus is present on the inside of the cell. ये एक याद रखने की बात है कि कार्बोक्सिल ग्रुप जो है, कार्बोक्सिल टर्मिनस जो है, वो सेल के इनसाइड होता है. In that, it is very difficult for a number of technical reasons to prepare crystals of GPCRS that are suitable for X-ray crystallographic analysis. जो कि ये क्यों ज़रूरी है कि we want कि X-ray जो है crystallography वो हमें इन प्रोटीन्स की बड़ी अच्छी कंफर्मेशन दे देती है, बट स्टिल इट इज़ डिफ़िकल्ट कि उनकी क्रिस्टल जो है वो प्रिपेयर की जा सके और एक्सरे क्रिस्टोग्राफिक एनालिसिस जो है वो मिल सके.